So next plant growth regulator is the gibberellin. So gibberellin after the auxin. The auxin our auxins are known as the plant growth regulator. And next one or the next category of this is the gibberellin. So gibberellin are the class of the plant hormone that play a very crucial role in various physiological processes such as seed germination, the stem elongation, the leaf expansion, and the flowering. Okay, so the biosynthesis, storage, breakdown, and the transport of the gibberellin are highly regulated process in plant, which we'll discuss in this lecture. So first, let's talk about the discovery and the significance of the gibberellin. So, discovery occurred, in the Japan, and the gibberellins were discovered by the Japanese scientists in the early 20th century, we can say. Okay, the rice farmer in the Asian country, they observed a disease which is known as a foolish seedling diseases or the Becan diseases, where the infected rice, they grew excessively very tall, but they failed to produce the seed. Okay, now when the association with the different scientists, they found, or the plant pathologists, they investigated that the Becan disease found in the excessive tallness in the infected plant was induced by some chemical which is infected, or which is secreted by some fungus, and later or that fungus was identified as Gibberella fujikuroi. Okay, then the chemical responsible for the tall growth, they were isolated from the culture filtrate of the fungus, and then the gibberellin and then the gibberellins were named after the name of this fungus. So this particular gibberellin, they play an important role in the plant growth. They were later found to be a plant hormone with a diverse effect on the growth and the development, the stem elongation. It includes the seed germination, flowering, and the fruit development. Okay, after that, the mutant with the altered responses to the gibberellin, such as the dwarf phenotypes in peas, it is controlled by the Mendelian inheritance. They have been instrumental in understanding the gibberellin biosynthetic pathway and identifying the biological active gibberellins in the plant. However, the discovery of these gibberellins and their role in regulating the plant growth, they had the significant implications of the for the agriculture and the plant sciences, leading to the advancement in the crop production and also understanding the plant physiology. So let's talk about the structure of this gibberellin. Okay, so this is the different gibberellins are found ranging from G1 to the G20, and the gibberellins are basically a diterpenoid, we can say. Okay, so these are the diterpenoids. Okay, that are formed from the 4 isoprenoid unit. We can see the 4 units are there. Okay, 1, 2, 3, 4. So these are the 4 isoprenoid unit. Each consists of the 5 carbon. We can see 1, 2, 3, 4, 5. Okay, and each consists of the 5 particular carbon. Now they possess the tetracyclic 4-ring structure, which is known as the N-gibberellin skeleton, containing the 20 carbon compound, and these terpenoids are compound which is made up of 5 carbon isoprenoid building blocks. Okay, so basically the gibberellins are the diterpenoids that are formed from the 4 such isoprenoid units. The gibberellin biosynthetic pathway, they can be divided into the 3 stages. Means each of the different stages, they residing in a different cellular compartment, which we will discuss in a while. So 3 stages will occur in the plastid, endoplasmic reticulum, and the cytosol. Now, as more the gibberellins from the Gibberella and the different plant sources, they were characterized. So the scheme were adopted to the number then ranging from the GA1 to GAN in the chronological order of their discovery. Okay. So some of the gibberellins, they have the full complement of the 20 carbon like this. 20 carbons are there, that is the C20 gibberellins, or the, we can say, other than there are the 19 uh, uh, C carbon 19 gibberellins are also found, because the one carbon is lost to the metabolism, and there are other variation in the basic structure, especially the oxidation state of the carbon 20 and the number and the position of this hydroxyl group. Okay, so despite the plethora of the gibberellins present in the plant, the genetic analysis have demonstrated that only a few are the biologically active as the hormone. All the other, basically, they serve as a precursor to form the inactivated form. And the basic structure is that this end coron or the end gibberellin structure, where the 20 carbon gibberellins are found. However, the gibberellin A1 is the most abundant and the commonly found gibberellin present in all the particular plants. So let's see or explore what are the biosynthetic pathway of this gibberellin. So the gibberellin biosynthesis basically occur in multiple plant organ, like the germinating embryo, the young seedling, the shoot apices, and the developing seed, and even in some fungi also. It is generally accepted that there are the three principal sites. We can say these are the three principal sites for the gibberellin biosynthesis. What are these? The developing seed and the fruits, the young leaves of the developing apical buds, and the elongating shoots, and the apical regions of the root. Okay, the immature seed and the fruits are the prominent site for the gibberellin biosynthesis, and this is based on the observation that the young fruit seed and the seed part, they contain the large amount of the gibberellins, particularly during the stage of the rapid increase in the size. In addition, the cell-free preparation from the seed, they are also able to actively synthesize the gibberellin. Now, the site of this gibberellin biosynthesis may be developing endosperm, the young, we can say, the cotyledons of the legumes, or the scutellum of the cereal grain, scutellum. Okay. So as the seed mature, the metabolism, they appear to shift in the favors of the gibberellin sugar conjugate, because gibberellin, they get associated with the sugar. So they shift toward the gibberellin sugar conjugate. Okay. Now it is not as easy to obtain the clear evidences that the gibberellin biosynthesis they occur in the root and the shoot. This is partly because the gibberellin level are much lower in the vegetative tissues. Okay, in the vegetative tissues, the gibberellins are very low in quantity. Okay, the vegetative tissues, they also yield the cell-free preparations that shows that they are the less active, suggesting that the enzyme level for the gibberellin metabolism, they are lower than for the reproductive tissue. So this is how the biosynthesis or we can say the synthesis of the gibberellin occur in the different part of the plant. So the three stages are there in the